Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter of class 12, that is the principles of inheritance and variation. In that, so many things we have covered, and in the previous lecture, we are initiated a new topic, that is allelic gene interaction, in which we have discussed already about the, we can say, incomplete dominance, codominance, okay, that we have discussed, okay, and multiple allele also we have discussed. Now today we are going to talk about lethal gene and pleiotropy. Okay, so first we are going to understand what is lethal. Actually, lethal means harmful. Lethal means harmful. So let's talk about lethal gene. Actually, lethal gene, there is no need to discuss it much more in detail, because it's not usually asked in syllabus. But we should understand. So, what is lethal gene? So, lethal gene, we can say, are the special type of gene, special type of gene which generally regulates, which generally regulates some character, but also affects but also affects the viability of an organism and and causes death in homozygous condition. Then these genes are known as particularly a lethal gene, and they cause this death in homozygous condition, that is known as lethality. Okay, so now it is known that lethality or death of an organism can occur at any stage, occur at any stage of life. And this lethality is of three types. It is of three types. And in these three types, the first type which is known, that is absolute lethality. Absolute lethality means death occur at embryonic stage. Death occur at embryonic stage before birth. Before birth. It is severe and rare. If we take an example of this, then hemophilia in human female show absolutely lethality. Okay, after this, the first type, absolute lethality, which occurred during the embryonic stage before birth. The second type, which we are going to study, that is sublethality. And sublethality means death occur after birth. Death occur after birth, but before maturity due to lethargy. Then this type of death is known as sublethality. If we take an example of sublethality, then sublethality is very common. And the example is most of human genital disorders like sickle cell anemia, etc. And third type is delayed lethality. Delayed lethality means that occur after maturity, means reproductive maturity. Then it is delayed lethality. Example is late age genital disorder like Alzheimer and Huntington chorea. So this is the different type of lethality. Now, one of the important fact is that lethal gene is discovered by Cunot in coat color in mice. Okay. Now, if you make a some cross, suppose in coat color in case of the mice, if you see there can be two traits are possible. One is yellow and one is brown. Okay, yellow is a dominant one and brown is a recessive one. Okay, so if we make a cross of this in the coat color, you will see that if you make a cross of yellow with the yellow, that is capital Y small y with the capital Y small y, then three offspring are going to be produced like capital Y, capital Y small y, and small y small y. This all are produced into 1:2:1 ratio. And when they are produced in 1:2:1 ratio, this one represent a condition which die in embryonic stage. So the ratio remaining of yellow and brown is 2:1. Okay. Same way, if we see The another cross which is discovered of lethal gene in snapdragon plant. The character which was taken, that is leaf color. Leaf color has two traits. One is green and one is golden. So if you make a cross of golden with golden, which is a dominant trait in heterozygous condition, then three types of offspring are going to reproduce. That is capital G, capital G, capital G, capital G, capital G, small g, and small g, small g. And this all in a 1:2:1 ratio, where this capital G1 die in early embryonic stage. The remaining ratio of golden is to green is 2:1. Okay, so ultimately the ratio of living is to death is 3:1. The ratio of living is to death in a clause is 3:1. If one is dying in homozygous condition and three are remaining in the terminal condition. But if we talk about ratio of lethal gene, then ratio of lethal gene present in that is 2:1. Okay, on the following previous clause we can say. Okay, so this is all about what a lethal gene. Actually, for lethal genes, many questions will not get. Okay, but you should know what do you mean by lethal gene. Okay. Now, next, after lethal gene, the thing which we are going to study, the another phenomena which show allelic gene and interaction. That is a pleiotropy. Now, in pleiotropy, if you talk, then the question is, what is pleiotropy? So, pleiotropy is a phenomena, and the gene which is responsible for this, that is pleiotropic gene. And this pleiotropic gene is responsible for multiple phenotypic events. So pleiotropy means multiple phenotypic effect. Okay. So if we talk about pleiotropy in more detail, then what is pleiotropy? The first question we can ask. So when a single gene may affect more than one character, means there is a gene is one, but it is affecting more than one character, then we call it as a pleiotropy or pleiotropy phenomenon. And occasionally a single gene product may produce more than one effect. So that single gene may be related to more than one character. So the phenomena is pleiotropy. You can see in this diagrammatic picturization that this is the chromosome, and in one chromosome, only this single gene is shown. This gene is responsible for trait A, trait B, and trait C. Means it is coding for a multiple trait or multiple character. Then we call it as a pleiotropic gene. Okay. So now we are going to discuss in detail about this. That what is this pleiotropic gene? Usually what happens, one gene, one polypeptide chain is produced, one gene, one code for one character, but here one gene is going for the many character. And if we need to understand how this one gene regulate more than one character, then we have to take some of the examples. And here we can take an example of for pleiotropy, that in humans, that is sickle cell anemia. If we talk about sickle cell anemia, it is a autosomal recessive disorder. Means whenever the autosomal gene is in recessive condition, then it is known as a particular autosomal and causing a disease in a recessive condition, that it is called as autosomal recessive disorder. Now for this, the gene are present on chromosome number 11, and the gene which are here, two types of gene are seen. One is HBA, one is HBS. Okay. So dear students, what happens here? In HBA, gene which is present on DNA, point mutation occur and converted into HBS. That is, it has a particular presence of sickle cell anemic gene. So what happens on DNA if we suppose the gene is CTC and this is the sixth codon. Okay, and here transversion occurs at the middle position. So the T is converted into A and CAC become at the sixth codon on DNA. So now if the transcription occur, you know that on mRNA, this code for GAG, and here it will converted into GUG. Now usually on polypeptide, GAG codes for the particularly glutamic acid. But GUG when code for polypeptide, it code for a valine. So at a sixth amino acid, this is what? Sixth amino acid in polypeptide chain. Here it is also sixth amino acid in a polypeptide chain. So when mutation occur in HBA, it converted into HBAS, that is sickle cell anemic gene, due to what? A particularly change in amino acid, a mutation in the sixth codon and change in amino acid at the sixth position of glutamic acid to align. So when the glutamic acid at the sixth position, it is producing a normal hemoglobin. But when the glutamic acid is replaced by valine at the sixth position, what will happen? The abnormal hemoglobin S type will be produced, and this is the normal hemoglobin. And this is abnormal hemoglobin. Okay, so this is the thing. Now, when abnormal hemoglobin is produced, the abnormal hemoglobin that is HbS in RBC, the oxygen stress will occur. And when in RBC oxygen stress occur, the cell sickle ship, actually sickle shaped RBC are produced that look like somewhat like this. When sickle cell RBC are produced, they are somewhat hard and rigid, so they get accumulated in fine, narrow blood capillaries. So circulation decreases. Circulation decreases. When RBC gets sickle cell, another effect is that they have a less lifespan. RBC die. So low RBC in blood, which causing anemia. Okay. One more effect of it is seen that when there has a sickle cell RBC, there is an increased resistance to malaria because Plasmodium parasite is unable to attach and penetrate sickle cell RBC. So this is the different effects can be seen by just a mutation in a single gene. So we can say the single gene is affecting many characters, so it is a pleiotropic gene. So in case of the sickle cell anemic condition, in a different individual, when there is two alleles that are HbA, then there is a normal RBC are produced and the individual is normal. But if it is HbA and one allele is affected and it is HbS, it is carrier, but symptoms are not seen. So we can say carrier but normal. They have a sickle shaped RBC and as well as normal RBC. Both normal and sickle shaped RBC are produced. When there is a HbS condition, both alleles are defective, then it causes sickle cell anemia. Okay, and this show sublethality as well as so the death after birth but before maturity. Okay, so this is the thing which occur in case of the particular condition. So if we make a cross of these, then F1 will be HBAA HBS. If we do selfing, then HBA HBS will cross with the HBA HBS, and three gene type will be produced: HBA HBS HBA 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 HBS and HBS HBS. Here these are produced in 1:2 ratio, and here this HBS HBS condition causes a death. Okay, in this condition the death can occur. So this is the effect of one gene when it controls many characters. Okay, so this is the particular example of pleiotropic gene, that is the sickle cell anemia. After this, next example which we are going to discuss, that is the starch synthesis in peanuts. Okay, and in the starch synthesis in pea seeds is controlled by a one gene. It has a two allele, that is B, U, V. Dear students, starch is synthesized effectively by the capital B homozygote, and therefore large starch grain is produced in the capital B homozygous condition. But in contrast, when there is a small b homozygous condition, have a lesser efficiency of starch synthesis to produce small starch grain. So if capital B gene is in a homozygous condition, capital B represents the large grains are going to be produced. But when there is a small b condition, smaller starch grain are produced. And if you see the after maturation of the seeds, capital B seeds are round, whereas small b seeds are wrinkled. Okay, so heterozygous produce a round seeds, and so capital B seems to be the dominant allele. But the starch grain produced are of intermediate size in capital B condition. When there is a capital B condition, intermediate size is there, but the seeds are going to be down. So if starch grain size is considered as a phenotype, then it is showing an incomplete dominance. And okay, see, first I need to make a cross. So if you make a cross, you will understand. Okay. Okay. See, we're taking another example to opioid, that is starch synthesis and shape. Actually, starch synthesis means starch grain size. You know that starch grain and size of three types: long, intermediate, and small, long or large. And seed shape are two, that is round and wrinkled. But for all this, the gene are only two. Gene is capital B and it has a two allele, only capital B and small b. So if we take a one capital B parent and the small b parent, capital B parent having a large or long sized starch grain, and they are round in shape, but they have a small starch grain in small b condition, and the wrinkled seeds are produced. Now they give rise to the F1 condition, that is capital B small b. This is the parental condition. This is the F1 condition. F1 condition is capital B small b, so round. This seed shape produces a round and wrinkled. A round in capital B condition, and capital B small b condition, and wrinkled in small b condition. So capital B small b, so it is going to be round. But here this is capital B, this is capital B small b, this is small b. Why? Because star synthesis show incomplete dominance. And this seed shape, so complete dominance. So here capital B small b is of intermediate starch and the round seeds. Now we carry out a selfing of this capital B small b. So if I perform a selfing, then capital B small b is going to hit the cross with the capital B small b. It is going to produce three genotypes: capital B small b, capital B, capital B small b, and small b. Capital B means large starch grain and round. Here they are intermediate and round. They are small sized and wrinkled. So you can see large, intermediate and grain are in 1, 2, 1 condition. But here this round are 3 and this is in 1 condition. So because of this, seed shape is showing a complete dominance because it shows a 3:1 ratio. But in large, intermediate and small size, you will get 1:2:1 ratio. So this 1:2:1 ratio shows what? Incomplete dominance. Whereas 3:1 ratio shows a complete dominance. So the start synthesis show incomplete dominance and seed shape show complete dominance. In spite of this, both characters are controlled by one gene. So now you will understand that starch synthesized effectively by capital B having a large starch grain. In contrast, small b homozygous have lesser efficiency in starch synthesis and produce a smaller starch grain. After maturation of seeds, capital B seeds are produced round and the small b are wrinkled. Heterozygous produce round seeds in a capital B small b condition, so capital B is dominant over small b, but starch grain produced are of intermediate size in capital B small b condition. So if starch grain size is considered as a phenotype, then from this the angle the allele show incomplete dominance. But therefore, starch grain considered as a phenotype. Starch grain size is considered as a phenotype, then it will be showing complete dominance. If we see the seed shape, then we show the complete dominance. Means dominance is an autonomous feature of gene or the product that it has information. Means whatever the type of product they will produce, that type of trait will be observed. Okay. So you can see this starch grain size capital B, they are large sized, and small b, capital B, small b, medium sized, intermediate size, small b, small sized, and phenotypic unit size large sized in capital B, capital B, small b, medium sized, and small b, wrinkled. So the question, if we talk, best example of codominance is blood group in humans. We have discussed in the previous lecture. Codominance in lentil plants show the seed coat pattern and size of the lentil. Okay. So shape of the seed and size. Pattern means shape and size. So 1 and 2 are there. What is the scientific name of lentil? So we remember it is a lentils culinaris. So that is the lentil. What is pleiotropy? A single gene affect a more than one character, then it is known as pleiotropy. Okay. So till now we have discussed already what your part of a pleiotropy. Now we are going to discuss another concept, that is a explanation of the concept of dominance. Dear students, after discussing this non-allylic interaction, we will discuss this topic in the new lecture. So in the previous lecture and today's lecture, we have completed with all the allylic gene interaction. Okay. So thank you.